review the structure of a sarcomere, which we've just gone through in our handout number 5, so I'll work through it fairly quickly. To remind you, the areas which we discussed include the A band. The A is just an abbreviation for the A in the first vowel in the dark band. To recall, we said it's the area of the sarcomere which you would find myosin and actin. Right? So we said just visually it's going to appear darker under our microscope. What we added to our handout number 5 was that H zone, which is the area of which band? The H zone is of which band? The A band, the dark band. Right in the middle, which line is in it? The M line. So the H zone is right in the middle of the dark band, and it's the part where there's just the myosin molecule in that midline, but the actin doesn't project down into the middle of it. And we learned that the M line, it is the center, but functionally, what's the role of the M line? We said to do what? What's the job of the M line? What's the job of the M line? What's it? Very good. It allows for a point of attachment for our thick filaments. We also identified the I band or the light band, and we said which contractile protein is in the I band? The thin filaments only. And which line or zone is in the I band? The Z disc, the Z band, the Z line. Okay, so the I band includes the Z disc or the Z band. So you might add that in there. It's the area. Where we're looking at. Okay. It's the part of the actin that does not project in the A band, and it also includes the Z line. Everybody catch that? We have another image of the sarcomere. If you want to make reference to figure 12.3 in your textbook, in the first part of the image, you can see just kind of an artistic representation, and the image below is an actual transmission electron microscope image to illustrate that this is legit. Here we got image data to support it. This is a really exceptional image. So to get something like this, probably someone had thousands of preps to actually get this kind of textbook illustration. But we can very clearly see the H line, or H zone, excuse me, around the M line. You can see a nice little Z band, but it looks lighter than the dark area. These next images, again, top left is another transmission electron microscope, probably more representative of what most scientists find when they do electron microscopy. You can see really clearly the Z disc or the Z bands. You can see really clearly the A band, and as well then take away the light area, the I band. And then if you look in the background, it looks noisy because in a muscle it's not just muscle fibers. It's, we can see here the sarcoplasmic reticulum, the T tubules, right? There's all kinds of organelles and molecules, and so it makes sense that the image would look a little bit congested like that because a muscle cell is so packed with different organelles. And in the image on the right, a light microscope image, you'll notice the detail is less because the resolution is lower. Magnification power is lower, but you can certainly see the banding. And this is the arrangement of the sarcomere is what gives that term striations to a muscle fiber. This is why the cells look striped, because of the altering of those thin and thick filaments in the sarcomere. We already covered the structure of the thick filament. We learned that the thick filament contractile protein is myosin. We said each myosin molecule has a tail and a head, and actually it's two of them as the image implied, but I didn't formally address. It's two tails, two heads adjoined to one another. We learned that the heads are also called cross bridges. To actually make the thick filament, we have each myosin molecule grouped with others. Just tons of them, for lack of a combination. Lots of them joined together, making this real huge macromolecule, complex structure. We also identified the binding sites for the myosin molecule. So go ahead and repeat this in the image on page 2 of your Chapter 12 guided notes. You can see the thick filament. On any of the globular heads, just label the 2 binding sites. What are our binding sites? What are the binding sites? I've got actin and, very good, ATP and actin. You do that for everyone on there. I'm not going to ask you to do that, but they would be present in all the myosin molecules. Any questions now at this point about the thick filament or a myosin molecule, the skeletal muscle? Let's review the structure of the thin filament. We learned that the thin filament is not just one protein, not just the contractile protein. It has also 2 regulatory proteins. So actin is certainly there. It's our contractile protein. And we know, what can we add there about actin? What do we know about actin? Yes, it's the contractile protein, but also what? Did I hear it? Excellent. It has the myosin binding site. So on your actin, it is the contractile protein, but it also has the myosin binding sites. Actin, it is a contractile protein, so it's involved in contraction, but we're just reiterating that it has myosin binding sites. While we're here, on the little thin filament section on the bottom of your handout, just draw a dark area of your thin filament on the actin. Draw a couple of them and label those the myosin binding sites, reminding you that they're covered up by which protein? Tropomyosin. Covered up right now. What we've learned is actin, in this image, the purple spheres are contractile proteins, but their binding sites are not always exposed. They're covered up by a molecule called tropomyosin, which we can see here, and we use the term filamentous. It's a filamentous protein. It's long, slender type of protein. And its job, as we can see, is to prevent binding when the muscle is what? When the muscle is relaxed. So tropomyosin is blocking those binding sites when the muscle is relaxed. We also just discussed in handout 5 that troponin is the 3rd protein of the thin filament, and it's going to be our regulatory protein for contraction. So what it does is it attaches to actin. It also attaches to tropomyosin, but it also binds to what? To calcium. And so when calcium's not readily available, it's kind of relaxed. It's not active. What we're going to add too is once calcium's available, it pulls back what protein? Tropomyosin to expose the binding sites. So although the thin filament is thin, it has more proteins in it. They're just anatomically smaller when compared to the thick filaments. Are you becoming comfortable with the structure of the thin filaments, or do you have specific questions about the structure of the thin filaments that I can address? All right. And again, I kind of just put everything out there, so we'll walk through the details. If we kind of step back a little bit to what we covered in lab last week, we learned that our skeletal muscles are under control of which type of nervous system? Skeletal muscles are under control of the somatic nervous system. And we can see a somatic nerve branching out, the axon terminals branch out to interact with multiple muscle cells. In lab last week, we coined those, right, the motor units. One nerve can control lots of different muscle cells. The image on the right is a transmission electron microscope image that's been colored, so we can easily see, right, see better what's going on. The top panel, what's this? What is this area? Oh, my hand blows. What is this? What is that? Very good. It's the axon terminal. The green area is the axon terminal, and we can see tiny little what? Vesicles that contain which neurotransmitter? Acetylcholine. Very good. We can see the little, if it was a nerve to nerve, we would call that a synapse. But here we're doing nerve muscle, so we'll call it a close. It's a neuromuscular junction. It's a junction between a nerve and a muscle. Call it a neuromuscular junction. The whole thing is called the junction. The term that was used called the motor end plate. The motor end plate is just the specific part of the muscle. Okay, so it's the motor end plate is specifically the muscle part, but if we're looking at the whole interaction, we can say neuromuscular junction. But you should notice that there is interstitial fluid between the two. So typical stuff like we learned with the nerve, neurotransmitter's got to be released, it's got to diffuse across the junction and bind with what? What's it going to bind with? Muscle. What part of the muscle? The receptors. What kind of receptors are there from lab muscle? Do what? The nicotinic receptors. We can't see them well. They'd be in the dark area. Very good. But I can tell that this is a muscle. Can you see the dark areas down here? That's taking the sarcomeres, not on the side, but turning and looking on the end. Okay, so little proteins look like rods, right, turned on their end. As I get close. See the neuromuscular junction. Got a couple of additional images to illustrate. Here's a nice scanning image. It's also been colored. You can see the difference between the nerve and the muscle. The nerve is colored what color in this image? The purple thing, the red things are the skeletal muscles. Here we can see some terminals branching there. All right, so we define this term called a motor unit, and we'll kind of elaborate it a little bit better now. And
I think you skipped the neurostimulus.
it may be out of order. Did I skip over? I think I might have. Oh, I did. Sorry. Thank you. Okay. So we concluded from the images that our skeletal muscles are stimulated by our somatic nervous system. And we learned as well from the images that they branch. And we learned the term where a motor neuron controls a muscle is called a neuromuscular junction. So we're no longer using the word synapse because we're not looking nerve to nerve, we're looking nerve to muscle. We good to go? All right, so then we showed the images. And then as well, from the images, we learned that a single nerve, because it can branch, it can control multiple muscle cells simultaneous. And the number of muscle cells controlled by a single nerve cell are called a motor unit. And so what happens is when one nerve gets excited, it causes multiple muscle cells to contract at the same time. And so therefore, instead of just contracting a single cell, we can actually move the entire muscle organ itself. The next bullet that I have on this handout or this presentation is that it says that our motor units are recruited asynchronously. What does that mean? Our motor units are recruited asynchronously. What does asynchronous mean? What does that mean?
Not together.
Not together. Not at the same time. Our motor units are recruited asynchronously, or not at the same time. So we can handle the word asynchronous, not at the same time. What about recruitment? We used that word before. Ever been recruited for something? Do what? Being selected, right? Very good. So our motor units are selected at different times. The purpose of this is, if you remember, we said our skeletal muscles can contract really quickly and really forcefully, but what? So the offset is what? What do they do? Skeletal muscles do what? Really quickly. They tire really quickly. Even just me pacing across the room like this, very, right, kind of not a big deal, right? But if I kept doing this for 10 hours, right? If I use the exact same muscle cells over and over with this very simplistic action, I wouldn't be able to do it 10 hours. How long do you think I might be able to do it? I use the same muscle cells over and over and over. How long do you think I could do this? 10 hours? 5 hours? Why not even 2 hours? Looking at minutes. How long I can maybe do this very simple activity if I use the exact same cells over and over. Because our muscles need time to rest and recover. Replenish calcium, replenish what else? What else are we going to use? ATP. Okay? And so what happens is our brain will select different motor units with repetitive activity. So with each step, if I'm not using the full muscle organ, with each step, say the first step, I may recruit motor units, cell number 10, 23, 15, whatever, in my muscle. Okay? And now, with the next action, those muscles should be relaxed, but in the same muscle organ, I can recruit maybe cell 2, 15, 65, whatever. And now, while the next set of muscle cells are working, the previous cells are resting. And the way we can do that is by what's called asynchronous recruitment of motor units. So in other words, not just one nerve cell controls an entire muscle organ. Multiple muscle cells control a muscle organ, and we can recruit different ones at different times to allow for prolonged activity, so we don't kind of tire out with very simple actions. And it's called asynchronous recruitment. This is visualized from your lab that we did last week. You used the image on the top left, which you look familiar, and then the image on the right is from your textbook. The one on your left I like a little bit better. You can actually see the nerves going into the organ instead of just kind of sitting on top of the muscle organ. But here, for example, in this muscle organ, looking at a tiny little bundle of cells, motor unit 1 is selecting 3 cells, motor unit 2 is selecting 2 cells, so for, say, that previous example, first step, maybe I recruited motor unit 1, get the whole organ to move, and then the next step I would use motor unit 2, activating these cells while the others are resting. Does that make a little bit of sense? Figure 1219 in your textbook illustrates that we can recruit motor units. If I need to do more action, more powerful actions, I need to recruit more motor units. Just like we did in the lab activity last week, that very simple activity, just raising your, flexing the muscles right on the top of your forearm to move your wrists. It required motor units. We had to use some muscles. But then we add the 1 to 2 pound weight. We still need to do the same action, but more forcefully. So we need to add motor units. The brain predicts this based on your past experiences. And so you notice that nobody had this crazy, like, I don't know, like they were trying to lift 50 pounds, right? You didn't see my arm just kind of flail out of the way. The body, I told you they were 1 to 2 pounds. Your brain predicted that and selected the right amount of motor units, so we had a nice, fluid little motion. It's really interesting how the brain predicts and determines exactly how much nerve excitation and motor excitation you need to do a movement, so we don't look weird and jerky and flailing or whatever with things like tying our shoes or walking or whatever the case may be. And so what we concluded is the more fibers that contract, the greater amount of tension can be generated by that particular muscle organ. And so we've already used the word recruitment about our motor units. And if one nerve gets excited, it's going to excite all of the muscle cells which it controls. If it's touching 3 muscle cells, all 3 muscle cells will contract. If it's touching 20 muscle cells, all 20 muscle cells will contract when that motor unit gets excited. And then the last bullet, I kind of identified. If you have more workload in your body, you're going to have to recruit more motor units to bring about the appropriate force and tension. And so movement's nice and fluid and planned looking. So if we look at organs in our body, what we can learn is not all skeletal muscle organs have the same motor unit size in them. So our muscle organs in our body that are do very precise movement, like our hands, the nerves that control them are considered to be small motor units. What that means is one nerve controls fewer muscle cells at the same time. Conversely, muscles that are used for power, like those in your legs and your arms, they have large motor units because one nerve cell can activate lots of muscle cells at the same time and generate really forceful type of actions. So the takeaway is there's not a set number of muscle cells which a nerve controls. It may control just a few. It may control a lot. The point is, what is that muscle organ designed to do? Are you all okay on that relationship? What a motor unit is, or how we can... It actually ties back into the central nervous system and the frontal lobe and all those things, which we kind of said our brain controls voluntary movement. This next image that is shown is summarizing what we've covered in lab, so I'll move through this fairly quickly, so I can just completely inundate you with it. But reiterating the relationship between the axon terminal and the skeletal muscle, and how the skeletal muscle is excited by acetylcholine. The primary thing to remember is if we zoom in at that area identified as the motor end plate, what is present at the motor end plate? What is it? I heard, but I didn't hear it. Just repeat it, whoever said binding site. Binding sites, and we call them what? Receptors. What kind of receptors are under skeletal muscle motor end plate? The nicotinic. The nicotinic cholinergic receptors. We know they're nicotinic because they're basically chemically gated what? Sodium channels. So when acetylcholine is present at a skeletal muscle is going to effectively do what? If we let sodium in, the skeletal muscle is going to depolarize. And so that's the big deal with the skeletal muscle. It has to first depolarize at that area called the what? What do we call this area where the binding sites or the receptors are? The whole thing is called the neuromuscular junction, but if we're specifically looking at the muscle cell where the receptors are located, the motor end plate. At a motor end plate, a muscle cell will depolarize when acetylcholine is present. And what happens is the rest of the muscle cell has those familiar voltage-gated sodium and potassium channels. So once the motor end plate depolarizes, the muscle can generate a familiar thing called what? What can it generate? An action potential. So an action potential can carry across the membrane, but we also learned that the membrane dips down into the cell. What is the area of the membrane that can conduct action potentials internally? That is called the, what is that membrane that projects into the cytoplasm? It is labeled for you, and you labeled it in lab. It's called the T-tubule or the transverse tubules. Okay. The transverse tubules, do we have that in handout 5 today? Tomorrow? Okay. Yeah, so looking at the first page of handout number 5, you can see the transverse tubule. What should you have about a primary function of the transverse tubule on there? It transmits action potentials to the cytoplasm because why? It has what about the transverse tubule allows its conductive ability? It has I mentioned it for you a moment ago. What allows a membrane to conduct an action potential? What does it have? If we're going to conduct something, we need what? The sodium channels, and they're gated, voltage-gated channels. Your transverse tubule can conduct action potentials because it has our voltage-gated ion channels in it, which allows for the change in potential. Can I repeat that? Role of the transverse tubules? Not shown in this image projected is the sarcoplasmic reticulum. That'd be neat if it was shown. And you guys know very well now. What's the role of the sarcoplasmic reticulum? Calcium storage. And it can store it and release it in the presence of and what can allow for the sarcoplasmic reticulum to release its calcium? An action potential. So it can release an ion when there's an action potential. So what must the sarcoplasmic reticulum have on it to allow for it to release an ion when there's an action potential? Voltage-gated calcium channels. Why don't you add that on there on your handout number 5. The sarcoplasmic reticulum does store calcium, but it releases it because it has voltage-gated channels, voltage-gated calcium channels. And then point C in question 1 we've already covered. We talked about myofibril. We understood its arrangement in the sarcomere, so we'll go ahead and move on from there. What I want, all of that which we discussed and just described is called excitation, contraction, coupling. It's linking the excitatory events of the muscle to the actual contractile event. So let's just review what we learned in lab and also from homework 5 and what we've been discussing today. Real quick though, got a couple of alternative images just showing at a neuromuscular junction the presence of those nicotinic receptors and how they're just chemically gated sodium channels. When the acetylcholine's gone, the gates will close. All right, so excitation, contraction, coupling, and if you work through the phase slowly, It's coupling or joining excitatory events with contractile events. Because up till now, we've really been kind of describing events that are more like a nerve. And we've got to link those to actions of a muscle cells. Muscle, right, the contraction within it. And so we're trying to link, my abbreviation AP is action potential. It's those events that physically link an action potential of the muscle cell with the contraction of the muscle. We know that an action potential will have passes, or you can write conducts, along the membrane, and then down the transverse tubules, down into the muscle cell. We also learned that the action potential affects the permeability of the sarcoplasmic reticulum. What we added is due to the presence of those voltage-gated calcium channels. Very good. And the calcium is released from the SR, or the sarcoplasmic reticulum, to where? Where is it released to? Where does the calcium go? It goes from the lumen of the SR to the what? Where does it go? The thin filament of the sarcomere. So in the cytosol. Very good. Goes in the cytosol, and right, that's where it combined with troponin of the thin filament. These events are summarized for you in Figure 12.8, and these are what I asked you to cover in your lab that you did Wednesday and Thursday in your homework 5. How the excitation triggers the release of cytoplasmic calcium from the SR. And what we'll add to it when we look at the actual contraction is the calcium flux in the cytosol that actually initiates contraction. But we can't magically contract without first exciting the muscle cell, releasing that calcium out of the cytosol. So that's why it's called excitation, contraction coupling. It's not as quick as you might have presumed before taking this class. We'll add to it as well. The calcium's free in the cytosol. The muscle is probably doing what? If calcium's free, the muscle is probably contracting. So the next thing the muscle needs to relax, calcium should go where? Out of, where should the calcium go? Out of the what? Out of the cytosol to back to the SR. That's all the calcium the skeletal muscle does. It moves from the lumen of the SR to the cytosol and then back in the lumen. Staying in the lumen of the SR when the muscle's relaxed. Muscle excites, boom, cytosol. Muscle relaxed, boom, where? Relaxing SR. Action potential out in the cytosol. Relaxation back in the SR. That's what it's doing. Okay. You guys already accurately identified, just repeating, that once that calcium is released from the SR, goes to the cytosol, and it combines with troponin of the thin filament. We also already kind of put it ahead that what happens is that troponin causes shape, changes its shape, changes structure, changes its function, so that it causes tropomyosin to physically pull back and expose those binding sites on actin for what? What's it exposing? Exposing its binding to? Myosin. And then, at this point, if calcium's available, those thin and thick filaments just bind. We haven't looked at any what? I haven't looked at any contraction. We're just looking at binding. So we've got to add a new level of something, which you guys identified is ATP. So we'll get to that. Yeah, but the first thing is calcium is in the cytosol and allows the thin and thick filaments just to touch or bind with one another. But then we need to ratchet. And so this is more of a review. We'll get into more, I guess not a review, but an overview, and then we'll add more detail in just a minute. So maybe in the margins, just in the statement where it says each myosin head binds and detaches during contraction, what I want you to add is when ATP is available. calcium allows for them to touch, but what we'll add in just a moment is when ATP is available, the myosin can actually bind and ratchet and gives the contraction. It generates tension, allows for movement, or we can say contraction. So we're not only just shortening the muscle, we're generating force to move the muscle cell and the muscle organ, right, ultimately. Shorten that sarcomere. If each sarcomere shortens, each myofiber will shorten, and then hopefully if there's a lot of myofiber shortening, the whole cell will shorten, okay, so on and so forth. So what we added, I didn't want to get too confusing, I'll come back to that image, is it requires which molecule? On the next page we said requires ATP in order to do this. And what happens at a sarcomere is we don't change the shape, we don't change the length of anything, nothing physically becomes, like the thin filament doesn't change, we don't add any molecules or remove any molecules or whatever the case may be. The relationship between the thin and thick filaments just change so that they overlap more and just become more packed, if you will, in that small space. And so we can do a very simple activity using your hands. Okay, your hands are going to be your sarcomeres. We can't clearly represent it exactly, but your thumbs will be your Z lines, so hold up your Z lines. You've got a sarcomere here. And your thin and thick filaments are going to slightly overlap, so just slightly overlap your fingers. And so the muscle is relaxed. We're going to action potential, calcium release, ATP, and now we're going to contract. Just move your fingers, right, move your thumbs closer together. And notice that the distance is less. What has happened to your eye band? It's what?
Smaller.
It gets smaller, and your thick band became, the thick band actually became bigger, more apparent. Okay? So the thick band, let's use that, dark band, let me use the right term. The dark band is small in the relaxed state, and then we contract, becomes really apparent. We just change the proportion between the thin and, between the dark and the light band. The point is, you don't see the thin and thick filaments like wrinkling or doing something crazy. They're just changing the relationship. They're overlapping each other more during contraction, which is illustrated in this lower image. We can see the I band in the blue, the A band in the pink yellow color, and then during contraction, you'll notice the thick filament, the dark band becomes more apparent, and the I band becomes less apparent. But we're just overlapping one. There's no jumbling or mixing or anything like that. In that case, the muscle is in the middle of contraction. In order to do this, it's called the Crossbridge cycling. So this is kind of where I left you hanging in lab. We said the muscle will contract. Now we're going to add detail about how the actual contraction happens. If ATP is available, it binds to which filament? ATP binds to what in the skeletal muscle? Where is ATP? Do what? The myosin head. If you remember on the myosin head, we drew the ATP binding site and the actin site. So it's the myosin cross bridge that actually gets the energy transfer into it. And so ATP can bind to this. It's catalytic. It splits ATP, so that we transfer what? We're transferring, we split ATP, we're releasing, we are releasing a phosphate, and functionally we're releasing energy. We're transferring the energy into the myosin, from ATP into the myosin cross-bridge. Inorganic phosphate is released first, and then what else is released? ADP. It's been studied well, so that first, make sure inorganic phosphate is first released. The order is known well, so we're going to acknowledge first inorganic phosphate is released, and then ADP is released out. So it's very sequential. Okay. when that happens, the myosin, because it's bound to actin, changes that head shape, and it brings that actin in towards the center of the sarcomere. Just brings it right in. Okay, so we're trying to shorten the whole thing. Collectively, that action is called a power stroke, and it's oversimplified in this image that I've shown, as well as in your guided notes. It's been if you remember the thick filament, right, there's billions of these myosin cross bridges, but we're just looking at one. So when calcium's available, the thin and the thick filament can bind. So it's just first binding. And then we'll power stroke with the addition of what? What note could we add here? Power stroke with the addition of? ATP. Here's the deal though. Once that myosin does its work, it's going to let go so it can do it again, or the muscle can relax. That letting go, or described here called detachment, is also regulated. The myosin cross bridge will not let go unless a new molecule of what can be added to it. What can we add here? More ATP. Okay. So make a note, detachment occurs when a new molecule of ATP is available. And then we can bind again if what is still there. It can rebind if what's still available. What allows for the binding? Calcium. Okay. It can rebind if calcium is available. And it can do another power stroke if there's more ATP, and it can detach with more ATP. But it will not bind again if there's no more calcium. So the power stroke happens after the excitation. Okay? The power stroke is part of the actual contraction. But first we have to have an action potential to release calcium, to allow for the thin and thick filaments to bind. And then we can see our ATP powering the activity. And so we already covered that a new ATP molecule added to the myosin cross bridges allow for detachment, and the myosin goes back to its kind of resting shape. What happens next depends on cytosolic calcium. This is summarized for you, this power stroke. Entire Crossbridge cycle is summarized for you in figure 12.7, so review it and the text around it. First showing ATP, we can see it split. Calcium in this image is the green thing. We can see the binding sites are exposed. First, inorganic phosphate is removed. We complete the power stroke, and then ADP is released. And now they're still bound, and so we need to detach, so we're adding a new molecule of ATP, and then if calcium's available, we just keep going through. As long as everything's available, it should function right normally. What questions at this point do you have about the power stroke, the role of ATP or the role of calcium in this interaction? Got a couple of, this one's good. We've got a couple of video links for you, and of course your homework 5 had some as well, but I'll show a different. In this case, we're looking at the myosin on the top of the screen, and on the bottom of the screen would be the actin. Troponin and tropomyosin are not shown, but we can see very well the cross bridge and the the motor proteins of the myosin, the little thing that'll look like feet, kind of ratcheting. And they'll just alternate. What color's the actin in this movie? A single actin molecule is colored green. The myosin crossbridge is the blue-red thing. Okay, when it's activated, ATP detach, split, inorganic phosphate released. Keep powering through. And then ADP. ATP causes detachment. That's all it's doing. Does that help a little bit? Hopefully. All right, we also identified in order for a muscle to relax, calcium's got to go back into the SR where it can be recycled, as we talked about. And everything kind of goes back. We're out of time for today. I need you guys to continue reading in Chapter 12 over muscle mechanics. To remind you, Chapter 12, we're looking at page 322 through 355. So hopefully you've gotten in there. If not, you need to catch up because you're behind. And then we'll get into Chapter 13 as well, Wednesday and Thursday. So reading Chapter 12 tonight and completing handout number 5. See you guys tomorrow.